In this video, we will discuss about T cells mechanical regulations at different stiffness of ECM. What are the intercellular changes occurs whenever the T cell experience the surrounding ECM either in the soft conditions or in the stiff conditions. So whenever the T cells experience this soft mechanical environment, what it will does is it will cause these YAP phosphorylation, that is S associated proteins, and this phosphorylated S associated proteins can interact with this IQ GAP1 and form a complex, and this complex then interacts with this NFTA NF81, which is phosphorylated, and thereby prevents this NF81 to enters inside the cells or enters inside the nucleus. And also similarly, this since the YAP protein is phosphorylated, it also won't be able to enter inside the nucleus, and thereby it does not cause epigenetic changes that will cause these suppressions that will cause or that finally results in the suppressed metabolism and proliferations, or we can say that the T cells are not activated in that case where they experience the soft mechanical environment, whereas when they experience the stiff mechanical environment, which is mainly corresponds to the disease conditions or cancerous conditions such as pancreatic cancers. In this case, because of the stiff environment, the YAP phosphorylation will not occur, and that will make the YAP in the free state, and it can enter inside the nucleus and cause epigenetic changes. And similarly, since the YAP is not phosphorylated, so it won't be able to interact with this IQ-GAP1 and forms a complex, and thereby this will cause the loosely bound NFTA1, and it won't get and this NFTA1 which are phosphorylated will be reversed or dephosphorylated through this CRAC, which helps the Ca2 influx, and we have discussed in the previous videos that how this Ca2 influx occurs due to one of the mechanical forces, and that will cause this calcineurin and calmodium complex formations, and that helps in the dephosphorylation of this NFTA1, and this NFTA1 can also translocate into the nucleus, and together this YAP1 and NFTA1 can help in the epigenetic changes in a way that will promote metabolism and proliferations of the T cells, and the T cells gets activated. So these are the different signaling pathways that are experienced by the T cells under different conditions, or we can say that how this ECM regulates the intracellular signalings within the T cells, whether uh, their mechanical and the mechanical properties of the ECMs in the T cells regulates whether it is soft or stiff. Both of them plays a different role towards these intercellular changes that occurs in the T cells. And here the stiff mechanical environments is mostly corresponds to activations of the T cells, because in the stiff environment the NF81 and the YAP1 can translocate into the nucleus and promote these metabolisms and proliferations of the and results in the promoted metabolism and proliferations of the T cells. So, and sometimes this conditions, that is stiff conditions, can also cause this tumor progressions in a way that when the tumor cells characteristics are being acquired by the T cells, and generally in the normal conditions, since the T cells observed a soft ECM, then they remain in this state, that is the suppressed metabolism and proliferation state, and that's the general homeostasis within the T cells. And how this YAP phosphorylated converted into the dephosphorylated using LATS12 is not yet been completely known, or we non known, or we can say that there is no general consensus has been made among the research community that how this LATS 1 or 2 dephosphorylated this YAP. So these are two different scenarios based on the physical properties of the ECM. If you need to understand more about this T cells intercellular changes observed because of the different stiffness of the ECM, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.